Hi, everyone. I'm Shannon Wyman, the Scientific Communications Manager at Keystone Symposia, and today I have the pleasure of speaking with Dr. Charlie Rice, 2020 Nobel Laureate in Physiology or Medicine for the discovery of hepatitis C virus. Dr. Rice will be the keynote speaker at the upcoming Keystone Symposium on Positive-Strand RNA Viruses, which will be held in Keystone, Colorado on June 18th to 22nd.
One thing that I've always enjoyed about this meeting is that you never really know what's going to be presented. It's, the positive strand RNA virus space is huge, and in plants and animals, and so it's always fun to just sort of go and sort of take it in because of the unpredictable nature of what you're going to sort of hear. I'll probably talk about some of our work on trying to understand more about the host factors and pathways that some of these viruses actually need in order to replicate. We've become very interested in that, and we've also become very interested in how the innate immune system has evolved to really have this sort of amazing ability to block the replication of both positive and negative and many viruses. And sort of how does interferon actually work? One of the attractive features of and also one of the challenging features of coronaviruses like SARS-CoV-2 is the huge size of the viral RNA genome, right? Which is like 2 to 3 times the size of sort of other positive animal positive strand RNA viruses. And that presents some challenges for sort of studying the virus and developing genetic systems, but it also means that this virus encodes so many different functions in order to sort of evolve to be able to replicate in different species. That there really are a gold mine of potential targets to go after. I think there are going to be a lot of compounds coming out that target these different functions, which, the advantage there would be that some of these are probably going to be very highly conserved functions, like the RNA-dependent RNA polymerase, so that these compounds probably are going to have very good efficacy against, variants that arise in nature, and maybe even, sort of more diverse and distant coronaviruses that are, potentially going to, cross species and emerge in the future. I think one of the lessons to be learned is that it's really important to maintain, a diverse portfolio of basic scientists studying these different viruses, even if they're not, the cause of the current pandemic, or even if they're, sort of not a major disease agent, because it's basically a lot of these kinds of studies. As I mentioned, I think that we were positioned to move into working on hepatitis C because of our work on yellow fever. But yellow fever, people would say in those days, well, why would you work on yellow fever virus? It's not a big deal, sort of anymore in North America, for example. We have a vaccine, one shot, lifelong protection. But trying to understand more about sort of the workings of that prototype virus, I think taught us a lot that was sort of useful for other flaviviruses, like dengue and West Nile, but also other positive strand viruses like SARS-CoV-2.